is the questions are kind of written in an all-encompassing way. So there are questions that are like, you know, now we know how, we're like breaking down the structures inside, you know, the plasma, we're going to go to plasma membrane, we were talking, you know, about cytoskeleton and stuff like that. So, and all these things are linked, and so the questions are kind of written in a way that you describe the link between them. You kind of put them together. So there are questions like that. Okay. By Monday, what I will do is I'll have an idea of how to separate it, and then we'll talk about, you know, how you can approach it. I don't think I'll have like a 2-attempt thing, unless maybe somebody wants to convince me. But because it is just the way it's written, I don't know how exactly to do that. So let me think. I guess we do have stuff that is new, right? We have some new things on the test.
So maybe that's enough reason to do the.
Finals suck anyways, and I know we're all having a terrible time, so we would love the second time.
That's a pretty good argument right there. Okay, let me think about it. Maybe you can help me kind of mentally process this. Maybe it's the new stuff, or
exactly.
That's the double attempt stuff.
The new stuff and the old stuff sucks. I mean, the content.
This is a great class. This is the stuff I talk about at dinner parties and stuff like this.
Over margaritas on 2nd of Mayo.
Well,
so yeah, let me thank you for that input, and let me figure out how to do it, and then I'll have a more concrete plan next week that I'll share. We're still going to lecture. So we have plasma membrane. Let me look at this list here. Extracellular matrix. Plasma membrane's like 20 slides. Extracellular matrix like 38 minutes of a lecture. And then next week we have, I probably made it a little bit long, but we have cell signaling and then cell cycle and cancer. Historically, I haven't tested over the cancer stuff, but we'll talk about it, okay? All right, so that's another thing. We will go where we can get to, and then, you know, if I have to pull something out, I can do that. Okay, so it's not so stressful for next week. Any other questions? There is a writing assignment to think about, and there is a quiz in week 16 also. How do I do that? Oh,
it's not out.
It's not posted now because I want to see how far we get, but it'll open up. The plan is to have it open on Wednesday and give you until the Monday to do it, okay, or Sunday. That way we can spend, my goal is to have Friday where we can just kind of review and talk about what the test looks like, okay? All right, so then let's go ahead and talk about the plasma membrane. Some of this is a little bit of review. In fact, I'm looking at this and I'm like, didn't we talk about this already? But it's a little bit more in detail, I think. And I think the reason I feel like, did we talk about this already? I think I just recently talked about this in Biology 1, is I think the reason. So we're all pretty comfortable with the plasma membrane, but we're still going to look at the structure again. We're going to review transport of molecules and endocytosis. So we did talk about trafficking in that, but we're gonna really focus in on endocytosis. All right, so whenever we're talking about the plasma membrane, this is one thing that really still gets some people, even myself. The plasma membrane always refers to the cell surface membrane. That's what we're talking about with plasma membrane. Because you know, you have your mitochondrial membrane, you have your membrane of your nucleus and all that stuff. So the plasma membrane is the surface. It's what surrounds the cell, and it's what is the barrier to the intracellular stuff to the outside, the extracellular stuff. Okay, so it's the boundary that separates everything from the extracellular environment. we have to remember that it's a selective barrier. It's a major deciding factor of what is going to be able to come in and what's able to go out. All of that stuff. It also is where we have our interactions between the cells. We've already talked about those adherens junctions and those tight junctions and stuff like that. But there is another layer of information we can add to that too. So this is just an electron microscopy image of it. Nice and high resolution, 20 nanometers the scale here. The intracellular environment you can see is very full of things, proteins, all kinds of stuff, right? And then the extracellular environment is different. So it's not really holding on to the heavy metal stain as well. So there's a lot of charged things in here that are interacting with the, we probably have never talked about this enough, but the way that these electron microscopy images are generated is, remember, it's an electron beam. So the way that we capture the beam or the information from the beam is we stain things with heavy metals like gold and things like that. And so here we are seeing retention of the stain or the electron, and then we have we see retention on the surface too. So and then we have this little gap in the middle. Okay, and what is that? So this is a lot of proteins and stuff inside of the cell, proteins that line the membrane on the inner side of the leaflet and the extracellular side of the leaflet. There are proteins that run through here, but we can't really see them with this method because the hydrophobic region excludes the metal ion, right? Because it's a charged thing, so it pushes it out. When you're staining your stuff, it won't stick to it. Okay, so it just sticks to the proteins or the charged things. All right, so we're going to focus in first on the lipids. We know what the complex lipid is, right? The glycerol, 3 carbons, 2 fatty acid chains come off of 2 of those carbons, and then you have a phosphate. But then we also talked about how there was some sort of organic carbon-based structure that can be added to the phosphate group, right? So it's not just a single complex lipid that makes up the plasma membrane. There's a lot of different ones that make them up. So we have, so like phosphatidylcholine has a certain structure, ethanolamine, serine, and acetal. These are specific structures. And I don't really give you the chemical structure of them, but this is just the variety of those things. And they make up, this is the typical percent of a mole of lipid membrane. If you were to grab a mole of the lipid membrane, this is about what you would get of each one. So the majority is phosphatidylcholine of the complex lipids. And then we have, next comes the ethanolamine, and then serine and inositol are in a low percentage. The leaflet that these are in matters too. So remember, it's a lipid bilayer, 2 layers. There's an outer layer and an inner layer. This I can't remember it necessarily for all of them, but for the phosphatidylserine, that's typically kept on the inside, the inner leaflet of the membrane. Okay, then we have cholesterol, sphingomyelin, and glycolipids. So a couple of other things that can be in there. Cholesterol is about, like if you were to add these up, you'd probably get about, between sphingomyelin and all these things, it's about 50% of the total lipid bilayer, and then the rest, and then it's about 50% cholesterol. So there's a mix, an even mix of the two. So this is just a picture of those organized in a little more detail. So here we have the outside of the cell, and we have our phosphatidylcholine out there. Ethanolamine, phosphatidylinositol, and serine are positioned on the inner leaflet, where they are. A glycolipid is basically, we have the fatty acid chains, but then we have a sugar. We have a sugar attached to that. Okay. These are only in the outer leaflet. The glycolipids are only in the outer leaflet of the membrane. isn't that interesting? Remember those glycosylated proteins and things like that? Where did they end up? When we trafficked them from the endoplasmic reticulum and the Golgi, if they were decorated with the glycosylation, they typically are on the outside. So that's another way to orient proteins in the membrane. Sugars go on the outside. All right, so again, cholesterol is about the same percentage as the others, and that's what this little carbon structure is here. Carbons and hydrogens and a little OH group. They are about 50%, and they're in both leaflets of the membrane. Remember that this is like a viscous oil, and it's self-sealing. So the way that these things work is the hydrophobic stuff stays together because it's really trying to get away from the aqueous environment that's inside and outside of the cell, right? So that hydrophobic binding or the hydrophobic interaction is what keeps these things intact. And if anything tries to come through, they're unable to because this really repels anything charged or anything like that. Now, if it's hydrophobic, it can make its way through, okay? Now, those cholesterols, oh my gosh. I was just telling somebody, it's like Murphy's Law. It's following me around. I can't just drop it. All the batteries have to fall out. Okay. We're here, we're together. Okay, so these back here, there is some, these are a little bit, they're more like an oil, and there's some movement in them. Cholesterols are a little more rigid. These are, you can't really see it here, but they're ring structures. And I know we're not biochemists necessarily, but ring structures are typically very rigid. They don't bend. But these fatty acid chains, they can bend, they can move a little bit. They can be moved. But these are a little more rigid. So they're interesting. I know I'm going to take you way back. I can't remember if we're going to talk about this in here. Probably not. But I know in Biology 1 we've talked about how these are the regulators of whether or not this is going to be like an oil or a butter. Okay, so you know how like fats can be, if they're fluid, their fluid state is an oil, and their solid state is like a butter, right? So these, when they are interdispersed like this, they can actually prevent these they can prevent the complex lipids from becoming more solid. If they really, really butt up against each other and they're not bent, or these chains are really straight, they can be a little bit more rigid, okay? But if you interdisperse the cholesterol, it gives fluidity to the membrane if the temperatures drop, okay? At the same time, if you pull all of these together, you can get these things that are a little more these like massive, more rigid little islands in the fluid, oily membrane. Okay, so these are called lipid rafts, and this is where we have a collection of, oh gosh, am I describing this wrong? Lipid rafts, cholesterols. Hold on. Okay, so it's not just cholesterol. I'm not describing it wrong. So these are, lipid rafts are high in their percentage of cholesterol. So we do have a lot of cholesterol coming together, but they're also connected with sphingomyelin. So sphingomyelin. And it just creates basically a section in the membrane that's recognizable by other things that are going to interact with the cell. And it also is something that kind of things can be locked into. Okay. They're kind of neat because they can it's a membrane structure, it's plasma membrane, but it's something that can organize maybe like signaling molecules that need to work together. They can be organized in a lipid raft. And they have an attraction then to the cholesterol and sphingomyelin nature of the raft. They are semi-solid patches, So they're a little more rigid than the yellow membrane that you're seeing around them. So we talked about how they're 50% complex lipid, 50% cholesterol, basically, right? They are, or it's really cholesterol to all the other lipids, because there was sphingomyelin and that in there. But then, of the total mass of the membrane, we have 50% of that complicated lipid mixture that we just talked about, and then 50% protein. And then we have our proteins in there. So the proteins are a lot bigger typically than those individual lipid molecules, whether they're cholesterols or the phosphatidylserine and all of that stuff. So it becomes, just to give you a perspective, it's like one protein maybe surrounded, or like the one protein is equivalent to 50 to 100 molecules of those lipids. So they're bigger. So we've already learned about our peripheral proteins, okay? So they're associated with the membrane through a protein-protein interaction, okay? They're interacting with a protein through typically not a strong bond, like not a covalent bond. The peripheral proteins would be interacting with like an ionic bond with some protein that's embedded in the membrane. Okay? The really key thing that I think it gets missed a lot for peripheral proteins is they are not typically associated with the membrane itself. Okay. It gets confusing though because we do have these fatty acid ones. So there are proteins that can be that can have a fatty acid chain attached to them. And right now. And so those, I think, those do fall under the realm of the peripheral protein. But so both are true. So we have the ones that are binding a protein, and then ones that are stuck into the membrane with a fatty acid group. So this is a prenyl group. It could be myristoylated. It could be palmitoylated. These words just refer to the fatty acid chain that is post-translationally added to the protein. All right, and then not to miss these, but we have our transmembrane proteins, okay, and our integral membrane proteins. That's what I'm saying wrong. I am saying that wrong. I apologize. So integral membrane proteins, transmembrane proteins. What is the difference? Does anybody want to take a stab at it?
Well, it looks like the peripheral membrane protein is attached to the integral.
So transmembrane means that they span the lipid bilayer with portions exposed on both sides. And integral membrane proteins means that they're inserted in the lipid bilayer, and they can be disrupted by reagents that disrupt the hydrophobic interactions. So it's labeling this as an integral membrane protein, both of them. I always felt that this was a synonymous thing. So integral meaning they can go through, they're in the membrane. Transmembrane meaning they're all the way through. So I'll have to, let me get better with that. We'll review that on Monday. Okay, so the common membrane proteins that are found in the membrane, so this is breaking that down a little bit more. You have the single pass, so they can go through just with a single hydrophobic region that fits in that hydrophobic area. So glycophorin is an example, and we have a single membrane spanning region. This is typically an alpha helix, so we've talked about this before. So alpha helix secondary structure. allows for side chains that are hydrophobic to be positioned so that they're okay sitting in that membrane. And it's a good structure to fit amongst the hydrophobic fatty acid chains. So we have a single pass, and then we can have multi-pass proteins. So band 3 actually passes through 14 times. Yeah, 14 transmembrane alpha helices go through the membrane. Notice again that the carbohydrates are on the outside of the cell side of the transmembrane protein. Okay? All right, so then here's another type of protein that is in the membrane, but not tetraspanning, or not spanning the entire membrane. So these are integral. These are integral. I did say that wrong. So here is one that is an integral membrane protein, and it is. So here we have a complex lipid, but we have a GPI anchor to a protein, to an amino acid. So we've got some sugars, but then they're added to a protein. We talked about how those are built before. So this is something that is tethered to the membrane because of the chemical nature of it. And then these here, so SARC and RAS, they have, this is an interesting way that it's done. They have a part of the polypeptide chain has a run of positive amino acids. And so they have an ionic interaction to the negative phosphate groups, right? So those phosphates have a lot of negative parts to it. So they have an interaction with that, but then, so that's one thing that holds it at the membrane, but then they also have, on the N-terminus of this protein, we have a fatty acid chain that fits right in there with the other fatty acid chains and complex lipids. All right, so then here is the RAS protein, and we have disulfide bonded fatty acid chains to those amino acids. So common feature for those integral membrane proteins is just to have a fatty acid chain added to it so that they can fit. They can fit in there. So something to point out about these is, in order to pull them out of the membrane, like as a molecular biologist working with a cell, you can use things that would disrupt the hydrophobic nature of the membrane. So we used to do assays where we would try to disrupt that stuff because we wanted to see if our protein that we were interested in was able to interact with another protein if it wasn't able to bind the membrane, because we knew that it had like a prenyl group or something added to it. So you could treat it with things. I'll give you an example of a drug, Philippin. Philippin is a drug that it doesn't disrupt the entire membrane, but it will kind of poke a few holes in the membrane at the right concentration, and it'll make some of these things fall out. Okay? If you were to lyse the cells, this is another interesting thing to think about. If you were to lyse these cells, spin down the contents, we talked about differential centrifugation, right? So you could spin out, start off by spinning out the mitochondria or something like that, right? In order to collect these proteins here, you have to collect the membrane. And that usually, so it's really hard to do that because these are kind of buoyant because they're like an oil, right? So in order to do that, you have to do it like in a vacuum at 100,000 G. And then when you get your membrane pellet, get all of the liquid off of it, you have to dissolve the membrane in like a hydrophobic solvent so that you can get the proteins away from the plasma membrane. So a lot of words just to basically get you thinking about what is the chemical nature of these things, how they're attached, why they can live in that hydrophobic region, and all of that. Questions? Okay, so now more details about the membrane. So the membrane isn't just complicated because it has different lipids in it, and it's cool because it has that dynamic where it can be very fluid or it can have parts of it that are very rigid. The proteins are stuck in them and all of that stuff. But another thing that is interesting about the membrane is that there are sides to the membrane. So the proteins that exist in one side are defined, and they're different from what might be in a different side. So let's see. So there are distinct domains. We've got our apical region that has the apical proteins that are there for whatever function that cell is. So that for the intestinal epithelia, you have a lot of proteins here that are involved in the absorption of nutrients, right? So binding and bringing in the nutrients, or maybe you have channels that are important for bringing in certain ions and stuff for that. On the sides, on the basolateral sides, that's the basal and lateral, this really refers to the sides and the bottom, and this is where you have, you're going to have those junctions, your adherens junctions and things like that, your hemidesmosomes and your focal adhesions. We don't typically have any of those up in here. You never have an adherens junction protein up there in a polarized epithelial cell. Okay, so the mobility of the plasma membrane proteins is restricted, and one of the major features that restricts the mobility of them, so they do diffuse, like the fluid mosaic model says that the proteins and things can flow through the little lake of oily lipid, right? They can flow around each other, around those lipids. But the amendment to that model is that they can flow to a certain border, and then they're stuck, right? So the major defining border is the tight junction. These stop any of the proteins that are living in the membrane here they're stopping them from passing into that apical region, and it stops the apical proteins from getting to the basolateral area. And they themselves are transmembrane proteins that they create this like woven network of transmembrane proteins that goes all the way around the cell. We've already talked about the belt of adherens junctions, right? And then just above those, typically we have a belt of tight junctions. So we have those distinct domains. So here is our picture of our plasma membrane. We've got a lot going on here. We have our transmembrane proteins. We have our lipid rafts. We see the glycosylation on the outside of the cell. The other thing, now you look at we did talk about this with our actin cytoskeleton, right? We have the spectrin actin complex, remember that, spectrin actin? And then it was like interacting with the band 3 proteins and stuff like that. Now we've connected all of that together. Those band 3 proteins, that was that like 14 pass transmembrane protein. All right. So we'll talk a little bit more about some specific proteins in the membrane, what they do. Lots of things that are done. You know, we have proteins that are sitting in the membrane so that other cells can recognize them, right? And there are proteins in the membrane that are, you know, helping them bind other cells and all that stuff. But then there are the proteins in the membrane that are regulating what is coming in and what's going out, right? So we have that selective permeability to the membrane. This is all review, I know, but we'll try to get a little more detailed in it. So we have, remember that crossing the membrane, nothing charged can go through without help. Now, if it's hydrophobic, it can go, ignore the transmembrane protein here, but it can go through the phospholipids by itself. So there are things that just pass right through. But here in this example, we've got glucose. Glucose is a big carbon ring structure that has a bunch of functional groups on there. It's bulky, and those functional groups carry charge. So the only way that it gets through is through a transport protein. Now, it can come through the process of passive diffusion if it's high in concentration on one side and low in concentration in the other side. So the spontaneous process is to go from high concentration to low concentration. Why? Because of entropy. So the second law of thermodynamics, right? So it will try to take up space, and it will pass, it'll go from this area to this area. The other thing to note about this transporter that you're seeing, this is facilitated passive diffusion. Facilitated refers to the fact that we're using a transport protein. Passive refers to the fact that there's no energy applied because we're using the second law of thermodynamics, and it's going high concentration to low concentration. Okay. The other cool thing to see is that we have it, the transporter is in this shape when it's picking up a glucose from the outside of the cell. Once it grabs the glucose and it binds certain side chains, amino acids inside, it changes, it's folding, and then it opens so that now the glucose has no place to go but to the inside of the cell. And then once this is expelled, then it can reverse its shape and go back to the other one. So that was a glucose transporter. There are also porins and ion channels. Water is a charged molecule, and one of the major channels that it goes through is aquaporin. So aquaporin is the channel that allows our water to move quickly through the membrane, and every cell has has a lot of those in the membrane. So the key thing about aquaporins is they actually will not allow, let me see, is one of these looking like one of them? The thing about aquaporins is they're impermeable to charged molecules. So it's basically an open pore in the membrane, but the amino acid side chains that are going to be in here are going to be such that they're going to be selective for what is allowed to go through, right? So not only is it like the chemical interaction that they might have with whatever passes through, but also the pore size is going to be a certain size, right? So it's almost like mechanical filtration for the membrane. There's a pore size that's going to be defined, and a molecule of only a certain size can get through. That's how channels typically work. Or a molecule of only a certain charge is going to be able to go through, or something like that. For aquaporins, they are impermeable to charged molecules, but they allow to other ions, but they are very selective for water. So H2O can go through. So in this case right here, we're actually seeing that this is a very general pore channel, and we're actually seeing a gated function. So you've got loading of some sort of channel cargo, and then there's an interaction in here where the gate gets out of the way. There's not a whole change in the overall structure of the transporter, but then it is open and everything's able to flow through. And everything's going to flow through in this direction as long as we have high concentration versus low concentration. So same thing, high concentration to low concentration for that stuff. All right. Any questions? So down here we have the combined efforts of the sodium and potassium pump and the K+ channel, the potassium channel. So in this case, we should note that typically the cell is kept at a high concentration of sodium outside of the cell and a low concentration of sodium outside of the cell. So it's 20 millimolar outside for potassium, and then for sodium it's 450 millimolar. It's quite a lot. All right, inside of the membrane, it's the opposite. We try to keep the potassium ions inside, so 400 millimolar, and then the sodium is 50 millimolar inside of the cell. This is something that helps us to maintain, basically maintain like a membrane potential. So notice that we have on the inside of the cell, around these pumps, we have a negative charge and then a positive charge on the outside. And that has everything to do with what's going to be interacting with those charged phospholipids. So if you have something that's interacting with the charged phospholipids, it's going to change the overall net charge of that region of the plasma membrane. Okay, in order to maintain this different gradient, we have the sodium potassium pump, and this works by grabbing for every two, I have to lock into my head these colors. So every two potassiums, we have that go in, we have 3 sodium that are pumped out. So we've talked about this transporter, I know, in other classes, but you have basically you have a binding site that is open to when it's folded in one way, it's open to the potassium on the outside, and at the same time it's open to 3 sodiums on the inside. And in order to move those, they have to be exchanged, basically. Exchanged to 2 atoms. This is against the concentration gradient because we're building these different concentrations across the membrane. So it is energy dependent. So ATP is required to make the changes in the transporter so that it can push these things in those directions. Okay? Our K+ channel is there at the same time, though, allowing for the exchange of potassium through the channel wall. So they kind of have to, there's a race between the 2 of them. This one has to keep pumping. And there's another avenue of movement. So most of our channels and porins, though, they do have a gate. So the gate can be maintained in the channel and not allow the potassium to move. So we can also have that. Questions? Was that review? Messy review? A little bit, yeah. Do you remember that? The sodium, potassium pump and all that stuff? Okay. All right, so how do these things work in nerve impulse? Oh goodness, I came in here and look at this. So you guys know what a nerve impulse is. Typically at the end of some nerve, you have some signaling molecules that are sitting in a vesicle. So, just linking us back to our other chapters. We have transcription of the mRNA in the nucleus, and then we have translation of the protein in the endoplasmic reticulum. We fill these vesicles with some sort of signaling molecule, right? That's how nerve the nerve impulse or the nerve signaling happens at the synapse or synaptic signaling. Okay. Okay, so over here, along the axon, along the length of it, we have these sodium channels and potassium channels, and they're gated, and they're opened in a way that propagates like the release of this membrane potential that is built by the sodium potassium pumps. Okay, so this is the way that the signal is basically passed along that long length of the nerve cell. Does that make sense? So by changing the ion composition, we're passing the information down the length of the cell. Okay? All right, once that is, so these are voltage gated sodium channels. So we're starting here at 0 milliseconds. They're closed at 0.9 milliseconds. This is after an impulse. Then we have the opening of the sodium channel and the regulation of the sodium concentration inside and outside of the cell. After that, at 2 milliseconds, then we go down the line and we have the opening of the potassium channel, and that's changed. And then at 5 milliseconds, there's resting. Okay? So once this is passed all the way down, then at the end of the cell, we have the communication is that these are pushed to the edge of the plasma membrane there. The vesicles are pushing the edge of the plasma membrane there. They fuse with the membrane, and then they release that signal. And then to continue on with working with channels, these are really cool because these signaling molecules are for ligand-gated channels. And ligand-gated channels, again, are the poor type of transport protein, but the gate is closed until their ligand binds them. You remember ligand is typically the word we use for a receptor and the molecule that it binds. So for ligand-gated, they bind, and then these channels open. And then we start to have a similar signal continue on the cell. Does that make sense? So you always think about like electrical firing of your nerves when you're thinking about, you know, the nerve impulse traveling through your body. And this is basically how that works. And the part that I think is difficult to maybe understand or see even in this diagram, I don't know if it's shown that well, but as we're changing that, what we are changing is this membrane potential. So you're changing the charge in the plasma membrane as you're opening and closing these channels. Okay? All right, so a lot of ion channels look very similar to each other in the way that the polypeptide chain associates with the membrane. They're typically multi-pass membrane proteins. So here's the potassium channel with six pass. And then here's the sodium channel that has, the way it works is we have all these 6 pass regions, and then they come together, you know, as going around so that there's a hole in the middle. And then there is the calcium channel looks very similar to that. So overall secondary structure, they might look the same, but their amino acid structure is going to be such that is selective for sodium versus calcium versus the potassium. They're also going to have different ways that they're regulated in their open and closed state. The gap junction channel is a 4-pass membrane protein, and then 6 of those 4-pass membrane proteins come together to make the board. But they all have those alpha helices in there. All right. Oh, here is a more detailed picture of our sodium potassium pump. So we have we're starting off with our sodium inside of the cell, but we want to get it outside of the cell. That's where we're maintaining a high concentration. We have, when it's open to this side of the cell, the active sites or the binding pockets are available. ATP is used in this way. We make a phosphorylated intermediate of the transporter. So we hydrolyze the ATP, cutting that phosphoanhydrous bond, right? And we're transferring the energy. We're releasing energy and then transferring a modification to the protein that changes the way it's folded. So then it changes and pushes the sodium out. Okay. Once the sodium is out, then these grooves are open to bind the potassium. The phosphate is then pulled off. So there's that transfer of energy again. So we're going to release that. And then we have our potassium inside of the cell. So it's an ATP-driven active transport process. Fun fact for your dinner parties, the sodium-potassium pump uses nearly 25% of the ATP. All that ATP that you generate in metabolism. Okay. 4 minutes left. I don't think I can get through 6 slides. Should we stop right there?
Yep.
Okay.